Welcome to our first Stages seminar, or I guess our second technically, Stages seminar for Cohort 3. Today our cohort is going to be sharing their individual research interests and topics. These are going to be very short presentations, and we will be recording. And we'll share those recordings after. And if you have any further questions for our cohort, feel free to reach out to them. Our first presentation today is actually going to be a pre-recorded one. One of our cohort is actually in New Zealand on her way to Antarctica as part of a 2-month research expedition. So Jerica is not able to be here today, but she did pre-record her presentation. So let me just make sure it's shared. Okay. And can everybody online see?
Yes.
Okay, thank you. And please let me know if you cannot hear, but we're starting now.
Hello, everybody. My name is Jerica Roselli. I'm a first-year CMSS master's student, and today I'm going to be talking about my thesis work, which is isolated versus organized convection around the globe using 10 years of GPM observations. Okay, so before I talk about what I'm doing with my research, I think it's important that I talk about what isolated and organized convection is and the differences between the two. So isolated convection is referring to individual separate convective cells or scattered thunderstorms that occur independently from each other. And these systems have a very short lifespan. Here's a little schematic of what this type of system would look like. So you can see that the updraft and the downdraft, which are the little red arrows, are not separated from each other. We also get no jet stream, no vertical wind shear, and the storm remains stationary. And like I said, these are very short-lived storms, and they are isolated, hence the name isolated convection. The picture there is an example of what this storm would look like in real life. So it's just like an isolated, random little pop-up thunderstorm. We see these a lot in the summer here. So these are the type of storms that I'm talking about when I talk about isolated convection. So organized convection is referring to a system of interconnected convective cells forming structures like mesoscale convective systems, squall lines, and tropical cyclones. So these are much bigger systems than the isolated systems, and they tend to last significantly longer because they're much more organized. So here's a little schematic of what that looks like. You can see we have a separated updraft and a separated downdraft. We have storm motion as well with these types of systems. And then we get a convective region as well as a trailing stratiform cloud region. So that's what that an example of what organized convection would look like. Here's an example of what that would look like in real life. So it's a much bigger storm than the isolated storm that we saw previously. And then an example of what this would look like on radar. So now that we know the differences between isolated and organized convection, we can talk about a little bit about the motivation behind my research. So basically, the role of organized and isolated convection in atmospheric circulation is recognized but not fully understood. This is due to the latent heating that is occurring within these systems. So we know that this is a primary driver of atmospheric circulation. However, it's currently not well represented in climate models. So that's what a major motivation behind this research is. I plan to look at these different systems and look at the differences in latent heating. The figure that just popped up here is just showing how important this is when it comes to heat circulation within our planet. So out of 100 units of incoming solar radiation, 23 of those units could be attributed to latent heat flux. So that's why latent heating is very important. So the goal of my research is to address the climate model gaps. We want to refine the climate models by improving our understanding in latent heating for both types of systems. So with that being said, my current research objectives are first to understand how isolated and organized precipitation systems contribute to the dispersal of latent heating. And I've done a little bit of work with that already. So you can see that in the figure that just popped up. I also want to look at where and when we observe isolated and organized precipitation systems. And you can see that in panel C. I've done a little bit of work with that as well. And then we want to look at the typical thermodynamic and dynamic environments in which these different types of systems occur. And I'm currently working on that right now. So I haven't done much work on that yet. For my methodology, basically I'm using the precipitation feature database that uses GPM data to classify, basically to distinguish where we're seeing any sort of precipitation features. So I do have specific definitions as to what makes these features organized or isolated. For the sake of time, we can just look at the little figure that I have. So in a 2 by 2 degree box, which is what we're using to classify these different systems, if most of the rain seems to be in one system, we're classifying that organized. Whereas if we have like scattered thunderstorms within that 2 by 2 degree box, that's what's making the system isolated. I looked at these different types of systems in the tropics as well as extratropics, and by classifying these events, I'm able to look at the differences in latent heating. In the previous figures you saw, I'm able to look at where and when they occur. So that's my methodology, and that's my research. If you have any questions, feel free to email me there. I do have access to my emails. And if you want to see more of the results that I have so far, you can scan the QR code to look at my results. But thank you.
I'm changing this to duplicate because it's a little bit difficult craning your neck to see what's going on up there. Okay, so our next trainee is Delaney Peters. Can you guys still hear on the microphone? I turned it down because I was getting feedback.
Yes.
This in my pocket. What direction?
Not sure.
I apologize if it sounds like I'm screaming at you. I talk really loud and microphones get kind of iffy.
Okay, so.
Can everyone see that online?
Yes,
And is it in presentation mode?
looks like.
Okay. No, I don't need presentation
Okay,
mode for
yeah.
them. That was what I was worried about. All right. Hi, everybody. My name is Delaney Peters. I am a first-year CMSS master's student in the remote sensing lab under Dr. Fay. And my research will be about investigating the planetary boundary layer using different data methodology techniques. But most importantly, we're going to be using GNSS radio occultation. And I'll get into the specifics here in a second. But first, everybody needs to know what the planetary boundary layer is and why we need to care about it. So the PBL is the boundary between the portion of the atmosphere in direct contact with the surface of the Earth and the free troposphere. So in this image above, you can see the area below the thin line in the middle is what we would call the Earth's surface influence. So we're looking at where the boundary layer is so that we can use it for weather and climate models, which depict different patterns and dispersal patterns. However, it's usually around typically to 1 to 2 kilometers, but it varies in different places. Now, why is it important? So we have to capture this accurately, the heights accurately, so that we can depict cloud formation, precipitation patterns, and pollutant dispersal. This is important for climate modeling because low-level clouds are very important when it comes to the heat budget. It'll foreshadow areas of drought or flooding, because if you have more low clouds, you probably have more precipitation and so on and so forth. And then how air quality will change with time, because most of your air pollutants are going to stay below the boundary layer because they can't escape into the free troposphere. So if that boundary layer is shallower or closer to the surface, you're going to have more air quality issues, and vice versa if it was deeper. So how are we looking at the PBL and the heights? So the first one we're going to look at is radiosondes, which are weather balloons that collect atmospheric conditions such as pressure, temperature, and humidity. The only bad thing about radiosondes is they're only collecting data from one place. The United States is kind of the only area that sends up radiosondes daily. The National Weather Service is how we get most of our data, but it's not global, which is what we really need to look at for climate purposes. So that brings us to the modeling. So the ECMWF reanalysis version 5 is a global model which kind of retroactively looks at atmospheric properties. So it goes back and it takes all of the radiosondes, the forecasting, observations on the ground, and it kind of puts them all together and says that this is what the weather looked like that day, so you can use it going forward. The problem with that is it's on a 0.25 grid by 0.25 grid, which means it's about 25 to 50 kilometers in area. It is high resolution when you're closer to the surface, but when you get higher up, it kind of shallows out because your pressure lessens. Now, we want to look at the Global Navigation Satellite System radio occultation because it's a spaceborne method, but it covers globally and it has very high vertical resolution. And I'll get into the specifics of GNSS radio occultation. So in a nutshell, basically there are your GPS satellites, which we use every day, and they're up there constantly. The NASA and UCAR, which is the University Center of Atmospheric Research, has low orbiting satellites that are following these GPS signals. And once those signals pass through the atmosphere, they bend. And from that bending, we can back out atmospheric properties, which will be helpful for modeling purposes. Now, when I say refractivity, it is a mathematical term that is made of 3 components, such as pressure, temperature, and your relative humidity. Radio occultation is already proven extremely valuable for higher atmospheric modelings, especially in hurricanes. But when I'm saying higher atmospheric, I'm meaning like 8 kilometers to 30 kilometers in the air. So it's kind of hard to spatially understand that because it's so high up. Now, why are we doing this? So we can use radio occultation to help improve the characterization of PBL structure. But we first have to validate what those atmospheric terms are so that we know how well the radio occultation is actually doing in the PBL. So to be able to accurately depict a wide variety of PBL structures, it's a top priority for the science and societal communities, which will help improve applications for weather, climate, and air quality. And these are some acknowledgements here, and I am open for questions. I don't know that I'll be able to see the online questions, will I? Yes. So at the moment, radiosondes is going to be global. So we're looking at our different regions kind of, so polar, subtropical, and then tropical. The polar one is Alaska because that is an ARM site, which they use high resolution radiosondes all the time. Our mid-latitude will be the SGP, which is Oklahoma. And then there are also field campaigns in Houston, so that can kind of get an idea of what the Gulf of Mexico looks like, specifically because that's where we live. And then the tropics will be in the Darwin area. And again, those are just because those are long-term radiosonde launch stations because of ARM, and that's where they. Correct. The data that I'm using right now, though, is past data. So I'll be focusing on 2021 because that's when COSMIC 2, which is the radio occultation site, has their most data. And then also 2007, because that's when COSMIC 1, which is the first mission, had the most data and the most applicable, if that makes sense.
We cannot hear what the questions are online, so if you guys
Oh, I
don't
will repeat them.
going
I'm sorry.
forward- That's
Yeah.
okay. Going forward, maybe the questions can be repeated so we know what the questions were.
Yes.
Thank
for sure.
you.
Okay.
Is that it?
And our next presenter is going to be Jasmine Nice.
Hey. Hello, everybody. My name is Jasmine Nice. I am a first-year PhD student here at A&M Corpus, and I am in the Movement Ecology Lab under Dr. Daniel Coffey. My dissertation is focused on the movement and trophic ecology of fishes here on the South Texas coast, so let's dive in. As a movement ecologist, one of the primary questions that we ask is why are animals moving and why do they migrate? So I'm just going to cover the overarching themes because they do relate to my dissertation. So the first of these is gametic migrations, which is our reproductive driven movements. We see this in a lot of animals that exhibit natal philopatry, which is when they return to the place that they were born to give birth. Another type of movement or migration is our alimentary migrations, which is seen in large pelagic teleosts such as tuna, where they exhibit vertical diurnal migrations at night to forage and feed. The last of these is climactic migrations, which is our environmental linked movements. We know this is pretty prominent right now, especially with our changing climate, but a lot of animals will experience range shifts or distribution changes because of changes in their environment, such as the white shark and many others that we know. So for my dissertation, I have 3 different chapters. The first of these is focused specifically on the movement ecology of southern flounder. We'll be looking at the spawning migration of these flounder and looking and seeing when that begins, when that ends, and how that overlaps with current management strategies. Just a brief background on the flounder, because this is the study subject of 2 of my chapters. This is an estuarine-dependent flatfish that is very important to commercial and recreational fisheries here in Texas. I don't know about all of you, but I love flounder. The problem is that because of overharvest and climate-related reasons, the spawning stock biomass has decreased over 70% in the last 40 years. So that's quite a cause for concern for us. And to study their movements, what we're actually going to do is we're going to tag them with acoustic tags. So we are going to surgically implant these into the body cavity of the fish. We're going to let that fish go after a short workup and hope, fingers crossed, that it swims by some of our acoustic receivers, where that tag inside of the animal is going to ping and the acoustic receiver will then record that individual. That's how we get our movement data. So this project is actually already underway, and hopefully we're getting some detections as we speak right now. We tagged 67 different individuals from August to October of last year. Here on the right is a map of our release locations in the red triangles and our receiver locations in the black dots. So it gives us a really nice distribution within our study system, and hopefully we get lots of good data on those guys. I'll also mention that we're working with the Center for Sportfish Science over at Hart Research Institute, so those are fantastic collaborators for us, and also how I'm doing my second chapter, which is focused on the trophic ecology and the contaminant analysis of southern flounder. So we're going to be looking at the primary habitats of foraging, the diet composition, as well as the level of contaminants present in flounder in our local estuaries here. So to do this, how we're studying trophic ecology, we're going to use 2 different methods. One of those is stable isotope analysis, where we will look at the nitrogen signatures, which will give us an idea of their trophic level, as well as the carbon isotope signatures, which will give us an idea of where they are foraging. We will also be doing a stomach content analysis to look at their diet composition and the microplastic assessment there. And again, thanks to those lovely collaborators at the Center for Sportfish Science, I have over 700 stomachs to analyze, so quite a few stomachs to literally sift through. And for my last chapter, I am doing something that is of personal interest to me. We're going to be focusing on a trophic ecology project of the local shark and ray community using stable isotope analysis. So the same method as before, just with a different group of animals. We're going to look at their dietary diversity, habitat use, shared resource use, and how that varies seasonally. So at the end of the day, why should you care about fish and sharks? Of course, for the first 2 chapters, I have the southern flounder. That big cause for concern is that 70% decline in the spawning stock biomass over the last 40 years. But more than that, we really don't know very much about their movement here on the southern end of their range here in South Texas. So that has not been studied explicitly. We're also going to be looking at the trophic ecology and contaminant analysis, and that will give us an idea of ecosystem health as well as potential implications for humans, because apparently we all eat about a credit card a week, which is pretty crazy. And then for the last chapter, the shark and ray chapter, we are going to be looking at a few species where Corpus Christi Bay is their nursery habitat, so a very important area for these species. And sharks and rays are very vulnerable due to overfishing, habitat degradation, and climate-related change. So we are looking at these animals, and it has not been looked at the community level. As I mentioned, this has not been examined. So it'll give us an idea of their resource usage, and then also those upper trophic level interactions, give us an idea of what's going on within the ecosystem. And with that, I will say thank you to all of my amazing funders and collaborators, especially at the Center for Sportfish Science and Texas Park and Wildlife, and then of course, our STAGES funders. And I will take any questions. Certainly. The question for everyone online is, were the flounder caught or were they lab raised? They were caught in the wild. We went out and worked with local charter fishermen this past summer and fall to tag those fish. Great question. Wonderful. Thank you.
Okay, our next presenter is Emily McGee.
Hi, everyone. My name's Emily. I'm going to talk about my thesis on the acoustics of common bottlenose dolphins when they are foraging. So before we dive into the details, I wanted to ask you guys, whenever you go to the grocery store, what of your 5 senses, what do you use the most to pick out your food? Does anybody want to answer? Yes, your eyesight. So whenever you're picking out your food, you obviously are going to look at what food looks good. You might even smell it to see if it smells good. Well, dolphins don't really rely on those senses. They mainly rely on sound to detect their prey and to forage. And so they have 2 main types of sounds. The first one are their echolocation clicks. I'm not sure if you can hear it, but this is an example. If you can hear that, you can tell that they're rapidly producing all of these clicks. And as they do this, they echo back to the dolphin and give them sonar information. So through these echolocation clicks, they can figure out what the object is, what kind of prey it is, what direction it's moving, etc. Their other type of sound are their communication sounds. This includes whistles. Dolphins have their own unique whistles to use for recognition, to maintain group cohesion, because they often hunt as a group. And so in the second image, you can see that they're working together to herd a school of fish. This is an example of a whistle. So dolphins need sound to successfully forage. So my thesis is looking at some unique foraging behaviors of dolphins in South Texas. In the first image, you can see that they are doing a foraging behavior where they follow trawlers. This is a type of commercial fishing that tows a net and catches things like shrimp or croakers or other types of fish. And so the dolphins will eat on the bycatch inside of the net or the discards cast out by the fishermen. And then the second image or the video on the right is a dolphin foraging along a seawall. This is called barrier feeding. It's basically using the seawall as a barrier to trap the fish. So both of these foraging contexts they make it easy for the dolphins to catch their prey, but there's quite a few risks associated with these different foraging contexts. So the red line in the study site image is the Corpus Christi ship channel. This is where all the trawling occurs. And The two yellow dots represent the seawall locations that the dolphins are often found foraging at. And this shipping channel is really, really busy. There's lots of boating activity. There's obviously the commercial shipping activity. This is where the trawling occurs. And so all of this human activity is occurring and causing increased noise pollution where these dolphins are foraging. And when dolphins are experiencing noise pollution, it can lead to something called acoustic masking. This is kind of like when you're at a restaurant and you're asking the waiter or waitress for an order and they can't hear you because the background noise is too loud. And so that can make things confusing. You could end up with the wrong order. And so this is kind of what dolphins experience when they are when there's too much noise pollution. The boat noise will occur at the same frequency that dolphin noises occur at. And so the dolphins can get confused, disoriented. They can even experience hearing damage. And so mitigating acoustic masking, understanding what the dolphins are experiencing are extremely important for their health, but also to be able to manage the environment. So going into my project, my first objective was to go out and record our dolphins. So I went out and recorded dolphins foraging behind trawlers and along the seawall last summer. And right now I'm working on analyzing their acoustic data. So I'm measuring what type of frequencies they're occurring at. And I'm also looking at the associated ambient noise levels. And so basically I'll be looking at if our dolphins are experiencing any type of acoustic masking. So this is my acknowledgements. Does anyone have questions?
If there aren't any questions for Emily, our next presenter is Cody Lopez.
Okay, hello everybody out in cyberspace, and our one audience member. My name is Cody Lopez. I work at the Center for Water Supply Studies, and I'm also a PhD student under Dr. Dorina Mergulet. So I wanted to give you guys a brief introduction to what my research is going to be about. Let's get right into it. So as a first beginning outline, I just want to talk about the overarching goal of my research, and then I'll tell you guys about my study site, the equipment that we're using to do this, and what I'm currently working on, and what I hope to learn from working with it. So, and our overarching goal of my PhD research is basically just to understand as much as I possibly can about the role of SGD, which stands for submarine groundwater discharge. So it's just what it sounds like. If you go underneath the water and you have groundwater percolating or being pushed out into the water itself, that is the submarine part. It's below water and it's groundwater just discharging into the bay. On our figure here, which is a pared down version of one from a paper Dr. Mergulet and I and Dr. Douglas are on, we can just see a couple examples here of SGD coming out of, I wonder if I can use the pointer on this. I'm not sure it's going to show up anyways. Of enhanced SGD, where we have some serpulated reefs or any kind of structure that's like rocky or sandy, can be a path of least resistance where groundwater is more likely to flow. Groundwater discharge can include things where water is just flowing directly out of the ground like that, or even if you can see that little expansion on the little figure here about wave action, when waves move over ripples in the sand, they push and pull water in and out of the sand. That's also part of groundwater discharge. And you can see this figure about the nitrogen cycle here. I don't want to get too deep into it because that'll be far too long of a presentation. But Suffice to say, groundwater discharge can bring in nutrients to an area, and the amount that it brings in, that flux, that amount that's coming into our water bodies is difficult to determine. So a lot of people just don't, and they kind of estimate it. Part of my research is helping to determine some of these values so we have a better picture of how much of this groundwater discharge plays an important or not important role in the nutrient budgets of some areas. Okay, so my study site, to study this groundwater discharge phenomenon, I'm working in the Baffin Bay area. There's 2 previous projects that have happened in this area, leaving us with a huge set of monitoring wells. You can see these purple diamonds represent some monitoring wells installed back in around early 2019, I believe. And then in our green data points here, these green circles here, we've got some data sets from wells installed in about 2020. So 2 projects going here. Putting in our wells. And with these wells, I'm going to try and study this issue in Baffin Bay. So Baffin Bay is a great study site for SGD with all of our monitoring wells because it has limited freshwater inflows. So it's one less variable to worry about. It's mainly just the bay water itself. It's a schizohaline bay, meaning that as the bay just sits there and evaporates, it keeps getting saltier and saltier until eventually some type of rainstorm like a hurricane comes and makes the entire bay a little fresher. The increased salinity limits the nitrogen cycling that occurs, so there's less things to worry about in terms of cycling. It has a known problem of high dissolved organic nitrogen. It's been recently well studied that there's some failing septic systems and failing sewer systems in the area, leading to an increase of raw sewage, which leads to this dissolved organic nitrogen in the area. Unfortunately, that leads to a lot of harmful algal blooms, and my role is trying to investigate these groundwater sources. What kind of role are they playing? How is it working? What's going on? To do that, we have these monitoring wells that are previously installed. They go about 15 to 35 feet under the ground. Each monitoring well has a pressure transducer inside of it, which is a little tiny probe that just records the water level of each well. And they take a reading about every 30 minutes. And we've had them in there since about 2021. So we've got lots of data to work with. We're taking these water levels and plugging them into some modeling. I'm working on modeling these groundwater elevations using as much external data as I can, things like the tide, the wind speed, what little stream flow there is. I want to combine as much of this environmental data as I can to get good and accurate models of what the groundwater situation is doing, because then I can use that to determine some groundwater fluxes, right? If I know what my groundwater elevations are, I can determine a basic estimate using things like Darcy's law of how much groundwater is going to the bay. And then as I learn more about those fluxes, I can apply what we know about the water chemistry from this groundwater to figure out what type of impacts we're having on our local bay, which happens to be Baffin Bay. Okay, that's pretty much all of what I'm going to talk to you guys about today because I'm hitting my time limit. So if you have any questions, please let me know. And of course, I want to thank my sponsors, the National Science Foundation, Stages, and of course, the Center for Water Supply Studies and the General Land Office. Thank you guys.
Any questions for Cody? Okay, and our next presenter is Jane G.
Hello, everybody. My name is Jane Ji. I'm interested in studying how disease impact predator-prey system over time. So I'm gonna share my interest in this order. The first one, impact of disease on predator-prey. When a disease enters the predator-prey system, several outcomes are possible. The first one increases predation on infected prey. So infected prey may become more subject to predation due to weakened health or altered behavior, leading to a reduction in the infected prey population. And then second one we can expect is indirect effect on disease dynamics. Predator can disease prevalence by, predator can influence disease prevalence by preferential preying on infected individuals, thereby reduction the number of disease carrier in the prey population. And then third is disease transmission to predation. If the disease can cross specific barriers, the predator can consuming the infected prey might contract the illness, potentially leading to the reduce in predator population. So considering all these chains of the event, I construct 4 differential equations in verbally. So to here, there are 2 populations. One is predator and predator and prey. I separate each population into 2. So one is susceptible and the other is infectious, prey and predator. So, and then I combine 2 models. One is Loca-Volterra model. It is very famous to find dynamic populations. And the other one is the SI model, which is one of epidemic models. So I have 4 differential equations. These account for birth and death and predation rate and reproduction and disease transmission and recovery rate. So when you see the one, susceptible predator increased by birth rate and then recovery rate, and reduced by predation rate and death rate and disease transmission rate. And if you see the second one, there is infectious prey. We assume only the infectious prey give birth only susceptible predation and prey. And then last 2 equations, there is no birth rate because predator reproduction rate is only tied to prey consumption. So this is verbal modeling, but the next one, I build 4 differential equations. So U and V represent prey and predator. And then S and I represent one is susceptible and I is infective. So to solve this 4 differential equation, I use Euler method. This one is very famous numerical analysis method. So my future work is identify the most important factors contributing to each population. And I use a random variable to observe population patterns. And then I also analyze the result when diffusion is added. So this is my references. Yeah. Thank you for your time and patience. Yeah. Do you have any questions? Yes. Equation 3. Okay. Yeah. Because susceptible prey and then infectious predator don't have, they are not increased by birth rate. They totally, it depending on how many they eat, the prey, that will be increase their numbers. If they don't have food, they cannot survive. So this, the reproduction rate the reproductive rate of the predator include the birth rate and then reproductive rate from the prey. That's why they don't have birth rate, yeah.
And you have a lot of parameters on your next slide.
Oh, yes, yes, it was. It was so complicated, so that was a very big challenge for us. But we figured out, yeah. I'm sorry, I can't.
So the questions that were asked by Dr. Tissot were in the third and fourth equations here, why birth rate wasn't included. And then the next one was lots of parameters and how to decide, and something to consider. Okay.
I have a question.
Okay.
Quick question. So I saw all of those equations and the different variables, right, that you consider, but are they all equally weighing on each one of those categories, or did I miss something? It's possible. Right.
Sometimes, yeah.
Go ahead.
Sometimes I use the same ratio. For example, predator and reproductive rate are the same. Or sometimes we use a birth rate, each is susceptible and the infectious are the same. So we try to simplify our variables, the parameters, yes.
But is that right? Like, is that possible that each one of them will impact in the same way?
But we changed the rate as well, yeah. So actually, we run the code. All parameters are different and all parameters are similar. We're just coupling some of them, the predation rate and reproductive rate the same, just like this. So first time we use the same parameter for predation and then reproductive rate, but next time we change the rate. So one is smaller and one is a little bigger or same. So we fixed some parameters and then we use different parameter to see the pattern of the population. Yes.
Well, I'd like to have more further conversation with you about how you're looking at this, because we're also using similar models, and it's just it's not always that each one of those parameters will weigh the same way, but I guess you're looking at how good your model outcome is. So, okay, thank you.
Okay. Our next presenter is Michaela Gwynn, who is actually returning from Cohort 1 as a peer mentor for Cohort 3. Okay. Hello, everyone. As Audrey mentioned, I was part of Cohort 1, so I am excited to be part of Cohort 3 as a peer mentor. And today I'll be sharing some of my dissertation research. I am in my second year of my PhD, and so my work is focusing on looking at the health of dolphins across the Gulf of Mexico. So I first want to start by introducing dolphins as bioindicator species. And essentially what that means is that the health of a dolphin can reflect the health of the ecosystems that they inhabit. And this is namely due to their high trophic position, their long lifespans, and their ability to accumulate pollutants in their tissues. And so what this figure is depicting is a concept we call biomagnification. And essentially, if you focus on the left side of this graph, we can say that a toxin or pollutant might be ingested by a low trophic level species. And as that organism is ingested in large numbers by higher trophic level species, the concentration of that contaminant can increase. And once it reaches our higher level apex predators like dolphins, it becomes a lot more toxic and in higher concentration. And so this is just one way that we can use dolphins as bioindicators of their environment. But there are many other biomarkers that are present in both the skin and the blubber tissue that we can extract to build sort of a health profile for these animals. So this will include things like stable isotopes, which are really informative of trophic ecology, habitat utilization. We can also use things like steroid hormones to assess major stress and reproductive events for these animals. Lipids and fatty acids are really good for looking at energy demands, overall body condition, and how that relates back to stress. Contaminants and pollutants, as I mentioned, are really important for understanding ecosystem quality and again, how that's impacting animal health. And then amino acids are really also important for assessing proper cell function and overall homeostasis in these quite challenging environments. And so the goal of my research is to use a combination of these different biomarkers to put together a comprehensive profile of health for bottlenose dolphins across the Gulf of Mexico. And so we have several sampling locations that we are focusing on. So here in the coastal bend, we have Corpus Christi and Redfish Bay serving as one of our sites. We also have the Upper Laguna Madre here in this area as well. And then moving a little farther north, we have samples from Galveston Bay. And then to the east, we have Mississippi Sound, as well as Mobile Bay, Alabama. And so the samples that we're getting from these different locations, some of them are going to be samples from live animals, some are stranded animals, and then we also have historic data going as far back as 2012. And so to collect blubber samples from stranded animals, right, when we find the carcass, we can take a large chunk of that tissue and put it in reserve for our analyses. But for those live animals that we're sampling in the field, we use a technique called remote biopsy. And so essentially we use a crossbow with modified darts to collect these small tissue samples from these free-ranging animals from our boat. And so we can then use these tissues and combine them with advanced instrumental analysis like a mass spectrometer to begin to build these physiological profiles. And so this is a depiction of what these profiles can start out looking like. And so this is essentially something you would get from a mass spectrometer. All of these peaks can be correlated back to concentration. And so really, when I say profile, I mean, we want to know concentrations. We want to know ranges of these different biomarkers and what they look like across different environmental contexts. And so my research is really important because a lot of these biomarkers are poorly studied. They might be studied for only certain demographics, only certain areas. And so we're really focusing on capturing a large environmental gradient that includes different water quality parameters, different age classes for the animals, different seasons, to really understand how they're adapting to their environment, how they're dealing with these challenges, especially in the face of global climate change. This is really important work to be doing. And so ultimately, this is going to help inform us of the current health status of these animals, and we can use that information for understanding how they're sustaining populations as these environments are continuing to change. And I would like to thank all of my funders, my collaborators, everyone in the lab, and we'll now take any questions. So for those online, the question from Dr. Tissot is, how difficult is it to get a sample of biopsied blubber from a dolphin using a crossbow? And the answer is, it is quite challenging, especially if you're not experienced. And so there's a lot of training that goes into this, and obviously only very well-trained professionals are the ones that are actually collecting these samples. In areas where humans are predominant, so like in Corpus Christi Bay where there's a lot of fishing and vessels are everywhere, the dolphins are really curious, and so oftentimes they'll actually approach the boat really closely, and that does help. But we do have to be parallel and at a minimum distance to collect that sample. And we need to know the group composition too. So if there's babies in that group, we don't want to be shooting at any of those animals because that could potentially be a really bad result. And so really it's just understanding the animals and how they're behaving in their group, and being able to predict their movements. And then a lot of times even they'll actually return to the boat after a sample is collected. So they're very curious animals, which is surprising. Yeah, so it's there's a lot of research on, you know, healing effects or uh, wound healing and things like that. And so, um, yeah. Okay. And our next presenter is going to be Sophia Mihalik. And in the chat, Evan Turner said, these are all super great presentations. So my research looks at the early life history traits of non-native and native damselfish in the Gulf of Mexico. So first, I wanted to do a little introduction to larva and larva dispersal because that's something that not a lot of people might think of or know about. So when reef fish have eggs, those hatch and they become larvae, and they're really small and they're not as developed and they're not fully a juvenile fish yet. So they don't have all of the sensory systems and physical systems to be able to move and eat. So they're very delicate. And I did some past research looking at benthic community composition, which got me interested in larva dispersal, thinking about how everything gets to where it is in the ocean, given the large size and large currents. So my current research will be looking at damselfish early life history traits. So specifically, I will be looking at the regal damselfish, which is pictured on the left. And they are non-native fish to the Gulf of Mexico. They are from the Indo-West Pacific, and they were first introduced in 2013. And so we don't know exactly how they impact the environment and how they impact other damselfish, because there's a lot of other small damselfish that have very similar niches to this fish. So my research will investigate how some of their traits during their early life period, such as when they're larvae, impact their competition between the species. And this is a photo on the right of our lab setup. So the tanks on the right will be for the adult fish, and then the larval rearing tanks are kind of right in the middle, and then on the left is some seawater. So why am I looking at the regal damselfish? As I kind of stated a little bit, the regal damselfish are non-native to the Gulf of Mexico. An invasive species is an introduced species that harms the environment. So technically, regal damselfish have not proven to harm the environment. There has not been any studies about it. So it hasn't been able to make that classification. So right now it's just non-native. But with its spread and its prevalence, its population is so large in some areas in the Gulf of Mexico, we do think that it could be harming the environment and harming native species. So that is why we are looking at it, because it was only introduced into the Western Atlantic in 2013 around the very bottom of this map. It came through the Panama Canal, and then since then it has spread all over the Caribbean and Gulf of Mexico. It spread there by 2017. So in 4 years, it spread extremely fast and its populations are only growing. What I will be doing is I will be looking at the otoliths of adult fish to analyze the larval period in the wild from these adult specimens. You can look back kind of like a tree ring on the otoliths and look back at their larval period, and the width of the rings is approximate to their growth during the larval period. And using wild specimens rather than lab specimens, we'll get that wild component and not be influenced by the characteristics of the lab environment.
And then for the larva, we're doing movement behavior analysis, tracking speed, direction, angle to really see their behavior and compare it among species. So some applications and why this is important. This could help manage the spread of the regal damselfish if we know its behavior and maybe what kind of food it likes or where it swims. It can help manage the spread and keep these ecosystems pristine and not have a new invader. This research can also be applicable on a broader scale and identify traits of successful invaders, because not every invasion is successful. Most species, when they're introduced to a new area, they just die. So the specific traits that differ from the native species that make these organisms able to survive in new environments and spread to new environments can be helpful to identify that on other species and identify potential targets of identify potential future invasive species, so then you can monitor them and prevent an invasion. Thank you to my lab and my PI, Dr. Joris, and this program. So if there's any questions.
Okay, if there are no questions, our last presenter today is Allison Gerard, who is another peer mentor. She was with us for cohort 2.
Okay, good morning, everyone. My name is Allison Gerard. I'm a second-year PhD student here at TAMUCC in the CMSS program and returning as a peer mentor for cohort 3. And I'm going to talk a little bit about my research. I study the nitrogen cycle in different environments, and I'm working on some method development. So I'm going to share a little bit about what I've been working on. So my project is sort of has two main purposes. The first is to develop these methodologies for studying nitrogen in the environment through stable isotope analysis. And I'm not going to talk too much about the method development today. If there's any analytical chemistry nerds here or online, I'm always happy to chat about this. But we want to measure isotope ratios of nitrate and ammonium in the environment. And we're going to apply these methodologies into very different environments that are both culturally and economically important. The first being an alpine ecosystem in the southernmost subrange of the Rocky Mountains. And we're also going to be doing a local objective looking at nitrogen pollution in an urban coastal watershed. So why study the nitrogen cycle? Of course, nitrogen is an essential element for all living organisms. And it's sort of interesting because there's tons and tons of nitrogen in the atmosphere and dissolved in the ocean in terrestrial environments. But most of this nitrogen is unreactive and unable to be used by living organisms to meet their metabolic needs because it's in the form of nitrogen gas, which is just two nitrogen atoms that have a really strong triple bond between them. And so it's not accessible for most living organisms. So we have to meet our nitrogen needs by using nitrogen in a fixed form, so where it's combined with other elements such as oxygen and hydrogen. And natural nitrogen fixation occurs primarily through microbial activity. So there's a small subset of organisms that can take that diatomic nitrogen and break it up and fix it with other elements. And so that makes nitrogen available to plants for assimilation, for their metabolism. And when we realized that this process was allowing nitrogen to become available in the early 1900s, this actually became really important for human society because we were able to develop nitrogen-based fertilizers to support agricultural practices. So that was really great. Human nitrogen fixation is probably why we're all alive today. However, this has dramatically altered the nitrogen cycle, and it's made a lot of this bioavailable nitrogen accessible in the environment. So now we have to figure out how are we going to manage all of this nitrogen in our environment. So like I mentioned, I'm looking at nitrogen cycling in two different types of ecosystems. The first is an alpine ecosystem. So you can think of these really picturesque mountains, high elevation areas where there's no trees, sparse vegetation, covered in snow half of the year. And because these environments are very rural, we don't expect large sources of nitrogen pollution that you would have in an urban setting. So most of your nitrogen is being delivered to these ecosystems through atmospheric transport. And once the nitrogen reaches these ecosystems, it can have cascading effects because these ecosystems aren't used to having lots of available nitrogen. So they have a low capacity to sort of handle the excess nitrogen in this environment. And we see things like biodiversity reductions, species composition changes, soil acidification, and so forth. So we want to figure out how is the nitrogen getting to these sensitive environments and what is the fate of that nitrogen in these ecosystems. And so we're going to use these methodologies to sort of characterize the nitrogen in the alpine environment. And we're going to do something similar in a coastal watershed, which has very different dynamics. Okay, so in a coastal ecosystem, you're going to have point sources of pollution. You're going to have urban runoff, agricultural runoff, but the purpose is sort of the same. We want to figure out how much nitrogen is there, how is it getting there, and what are the ecosystem effects? And can we link the sources of nitrogen to changes in the environment? And that's really important for things like mitigating this excess nitrogen, handling it once it's there, so on and so forth. And so I'll be using stable isotope techniques. When we know the isotopic composition of our nitrogen compounds, we can get an idea of where it comes from. So we see this plot here. This is just sort of a generic plot of oxygen and nitrogen isotope data from different sources of nitrogen that we expect, such as from atmospheric emissions or from fertilizer application and so forth. And, you know, for an example, we can use this to find some nitrate in our surface water and see where that came from. Did it come from runoff? Did it come from fossil fuel combustion? But the larger goal here is to develop those novel techniques to improve some of the shortcomings, I guess, of the traditional conventional methods and apply those techniques to measuring nitrogen in these ecosystems. So thanks everyone for listening. Thank you to Stages and NSF and everyone else, my advisors. And I'm happy to take any questions. Oh yeah, for sure.
Fairly different between mountain environment versus the coastal environment. Sample a lot during the year, or
Yeah, so my local objective, which is going to be Corpus Christi Bay and Upper Laguna Madre, that will be really high temporal resolution. I think we're going out like once a week taking water samples and groundwater samples. And that will be for a whole year. So we'll sort of capture a year there. For the alpine ecosystem, it's totally different. So much of your nitrogen and your hydrological cycle is like influenced by the snowfall, right? Snow cover, snow melt. So for that, I already did the sampling, and I did like an early season sampling when there was still snowpack, but it's like just starting to melt and supply down to the watershed. And then I went at the end of the season when all of that snow melt has sort of flushed through, and then seeing basically like what the nitrogen export is from, okay, accumulated in the snowpack over the winter, how did that get transformed throughout the growing season? So I have sort of dealing with the seasonality in that way for that objective. Yeah, I have concentration data and I have some isotope data from like stable or from IRMS, which I haven't dove into all the way, but the concentrations were really high at the end of the growing season. But I also happened to be out there during a rainstorm. So I expect that when I get my isotope data, it's all going to be from deposition because it was like raining the whole time that I was sampling. But that's different. Sometimes you expect to see high concentrations at the beginning when you have that snow melt because it's all being supplied down, not just through what is melting from the snow, but also from biological activity under the snow. It's all being sort of exported down. And then by the end of the growing season, you might expect to see really low concentrations because that's all been assimilated. But we see sort of the opposite because, and I think because of when I was sampling during a rainstorm. So the isotope data is going to really tell the story. Thanks so much.
Oh. So, our trainees were all given an assignment for today. Introduce us to their research in 5 minutes or less, and they all knocked it out of the park. No one went over that time limit, like by super amount, maybe half a minute, but, give yourselves a big round of applause for that. You met the goal of the assignment here. Great job, guys. And since we have a little bit of time, or if we have a little bit of time, because I know we're at time, I would like to give our stakeholders who have joined us online a chance to introduce themselves. So if you're able to unmute, Peyton, if you'd like to start.
Hi, everybody. Yeah, I'm Peyton Campbell. I'm the Air and Water Monitoring Coordinator for Coastal Watch Association. We used to be called Ingleside on the Bay Coastal Watch Association, so you might recognize that name. And we were founded in 2019, and we, to promote the health, safety, and quality of life of our residents and surrounding communities, we've just been doing partnered research with universities and independent community science to help further our environmental goals. It's good to see you.
Okay. Thank you, Peyton. Polly?
Hi, I'm Paula Hayaski. I work for Texas Parks and Wildlife, Coastal Fisheries Division. The main focus is basically the conservation and sustainabilities of Texas's natural resources for the present and future generations to come. So a lot of the everyday stuff is just thinking of how we can do these research projects to help benefit the fisheries management and everything.
Thank you, Polly. Polly is also a TAMUCC alumni of the FAMA MS program. Jason?
Y'all, this is Jason Pinchback. I work at the Texas General Land Office in the Coastal Resources Program. And my colleague Lucy Flores is also on the call. Together we work in the Water Resources Management Program area, which includes Texas Beach Watch, Texas Coastal Ocean Observation Network, or TCOON, the Coastal NPS Program, and Clean Coast Texas. So we're using these programs synergistically together to better understand changes over time and space. On water quality, our peak focus has been related to the fecal indicator bacteria and Enterococcus, and wanting to report those in a timely and accurate manner for stakeholders. And then beyond that, to try to understand why certain areas are behaving in a particular way, especially when we have higher advisories and higher risks for contracting an illness in a particular area. We're also working with Dorina and Dave Felix, some other folks on this call, and we have numerous research studies ongoing, and lots of fun. So we really appreciate y'all's partnership.
Thank you, Jason. Nick?
Hello, my name is Nick Carr. I'm the Science and Operations Officer at the National Weather Service office here in Corpus Christi, Texas. Some of you may be familiar with what we do, but we're responsible for issuing watches and warnings for a large portion of South Texas. Your typical weather watches and warnings, severe thunderstorm, tornado, winter storm, etc. We also produce forecasts for aviation and for the general public as well. We do a number of outreach activities related to weather and weather safety. And so, yeah, we've had partnerships in the past with TAMUCC for a number of joint projects, and we always look forward to having the opportunity to work with students and kind of, we call it research to operations, get some of the latest research from academia into operational meteorology, weather forecasting, and warnings.
Thank you, Nick. And finally, Evan. Yes, also a TAMUCC alumnus of the CMSS PhD program.
Hold on, having computer problems. Sorry, guys. I'm having computer issues over here.
Well, we can hear you, so if you, even though we can't see you yet, if you want to proceed, you can.
Sorry. Yeah, sorry. You're about to have to skip me real quick. Sorry about that.
Problem is you're the last.
Oh.
So if you would like to talk to us, we'll see you next time.
Yeah. You can hear me?
Yeah,
you can
very
actually
well.
hear.
Yeah.
Okay. Yeah, my computer's, it's like completely frozen, but all I can do is hear. So I'm Evan Turner. I work at the Texas Solar Development Board, and I've been here now for almost 8 years. It's been 7 years. I was at A&M in the early 2000s. I did a computer science degree. I left. I worked at the Texas Advanced Computing Center for a few years, and then I went back and I got my PhD in the CMSS program, and that was under Paul Montagna. And then now I'm at the Water Board, and it's a state agency, and it's pretty neat. It's sort of like a bank, and people, well, cities will come and try to get a bond for wastewater improvements or water improvements, and you might hear about that stuff. It's on the news to say, hey, there's a bond for doing a water improvement. Well, when you get the bond, the money doesn't come out of thin air. They have to get the money somewhere. So what they do is they come to the Water Board, and they get very low interest rate loans from us that are subsidized. But we also have a really big planning group that plans these water infrastructure projects, and then there's a science group, and I work in the science group. So right now I'm a data scientist in the surface water division, and one of the big things I do is work on our websites and our data products, doing collections and making sure that our websites have really good data. And that's one thing that we kind of pride ourselves here at the Water Board, is we're kind of a way station for connecting water data to stakeholders around the state. And the water data is coming from either us, or it's coming from USGS, NOAA, different partners all around the nation. So that's one of the things that I do here, and it's very excited, and I was really enjoyed the presentations. It's so neat to see what all y'all are doing, and man, I think y'all did just a bang up job. It's really hard to wrap what you do in a very short presentation, and I was very, very impressed at your timing. How everybody was able to get everything done so quickly and so nice.
Thank you, Evan. And thank you to all of our stakeholders that joined us today. We will be in touch throughout this next year, and hopefully we'll see many of you this term for mini workshops. So I will be reaching out about those again soon. But that's what we had for today. So another round of applause for our trainees. Oh, did you have a question, Nick?
Yeah, I have a question while you're here about the mini workshops. I know you gave us a range of dates. How many of those dates we need to be available for to do our little presentation portion?
To do your presentation, just one date, and then you are welcome to attend as many of the others as you are able to see what the other stakeholders' needs are for potential collaborations.
All right, thank you very much.
All right, so I hope everybody has a good rest of your week.